Thank you for that introduction, and I would like to express my opportunity to the conference organizers for the chance to speak today. It's a pleasure and an honor, and I look forward to some interesting discussions on the topic and sharing some data with you. My presentation title is Maternal Nutrition in Beef Cattle, Developmental Outcomes in Offspring. Developmental programming is a theme of which we're talking about today, and it's not a new concept. Developmental programming has been around in our minds for a long time. If we look at the Newsweek article, Where Health Began, that's from 1999. And if we fast forward to the Time magazine cover page, How the First 9 Months Shapes the Rest of Your Life, that's from 2010. It's 10 years later than the Newsweek article. Certainly from a human perspective and also from a livestock perspective, this has been in our minds for a long time. If you fast forward another 7 or 8 years and look at the Animal Frontiers, which is the review magazine for animal agriculture, published by the American Society of Animal Science, the entire issue was dedicated to developmental programming. What mom eats matters. And of course, the focus here, excuse me, was in livestock. If you move forward again, another few years to 2019, Animal Frontiers headline again, farm animals are important biomedical models. And one of the interesting things to me about doing work in developmental programming is that your data applies to livestock. I'm an animal scientist, and I'm proud to be an animal scientist, and I love doing work in that area. I think feeding the world and providing a nutrient-dense food is a very important thing to do for the human population. But also, our data can relate to human biomedicine, and certainly large animals and farm animals have been very important biomedical models for a long time. One of the things I also like about working in developmental programming is I get to address major societal issues while at the same time studying things I'm exceptionally interested in, like ruminant nutrition and developmental outcomes. Things like obesity, metabolic disease, and cancer, these things seem to be related to developmental outcomes and can be impacted by perturbations during pregnancy. Both nutritional stresses and other stresses seem to affect these things in offspring, even into the second and third generation. But also a major societal issue, in addition to the biomedical ones that I mentioned, are also feeding the world population. Um, World population projections continue to go up, and we're going to need a lot more nutrient-dense products. The projections for the increased needs in animal protein and animal protein products, as well as crop products, are very, very high. What's interesting is we need to make sure what's interesting and challenging is we need to make sure that we do that in a way that's balanced, in a way that's sustainable, in a way that keeps all sorts of things in perspective, because we want to make sure we provide for this generation of nutrition, but we also want to provide for the next and future generations in a way that protects the environment and protects our ability to continue to do things in a sustainable way. This is a cool thing about developmental programming because it, 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 it plays right into those areas. So maternal nutrition and developmental programming. Some concepts here are probably in most of our minds that inappropriate maternal nutrition during critical windows of development can result in altered offspring outcomes that persist into adult life and even cross-generationally. This developmental programming is driven by epigenetic mechanisms, most likely, and differs depending upon timing and insult of the sort of stressor, whether it be maternal nutrition or other sorts of stressors. Developmental programming and epigenetics. Epigenetics seems to drive developmental programming, and two of the major components of the epigenetic code are DNA methylation and histone modification. And these things are influenced by epigenetic modifiers or one carbon metabolites. And as nutritionists, we know those things as nutrients like methionine, folate, B12, choline, and some others that would influence one carbon metabolism that would drive into DNA methylation. So DNA methylation and one carbon metabolism, very important in terms of epigenetic modification, also histone modification. MicroRNAs are also involved in epigenetics. And I've provided 4 references here spanning 10 years that will provide some background of that for anyone who wants to do some additional reading. If you want to look at the December 2021 issue of Animal Frontiers, this whole issue was focused on the prospects for exploiting epigenetic effects in livestock production. And that's developmental programming effects, an excellent issue, and I'll point your all's readership to that issue. In developmental programming and animal models, we've used several different types of animal models, both bovine and and sheep. At North Dakota State, we've looked at various models from macronutrients to micronutrients. Early on, when we were studying this, we looked mostly at mid to late gestation models of maternal nutrition. We also explored some evidence, explored some questions in preconception. I've got some data on that, won't show it today. We've got an emerging wing of work led by Dr. Carl Dolan about what about the paternal effect. And on the maternal side, for the past 8 years or so, we've been pushing into this early pregnancy area. And there's some real reasons for that. And so, why early pregnancy? Well, one, cell growth and differentiation is going at an exceptionally high rate on a per cell basis. We go from basically single cells to a whole lot of cells and organ systems in approximately 40 days in beef cattle. Now, all those organ systems are not functional, but those organ systems are certainly identifiable, and those are massive, massive changes. Um, During early pregnancy, small changes in nutrient supply may represent bigger proportions of nutrient demand. Early pregnancy is the time of greatest embryonic loss. Conceptus nutrient requirements during early pregnancy have not been well established, and there's limited data currently available on uh, maternal nutrition effects on developmental outcomes when applied during early pregnancy. There's potential major impacts on offsprings and their outcomes. So if we take a quick look at the first 50 days of gestation in beef cattle, you can see on your left that we have day after mating starting at 10 and going across the timeline to day 50. You can see that we have maternal recognition of pregnancy, and we have a transient beginning increase in uterine blood flow, and we have elongation and attachment and sustained increased uterine blood flow. And when we get out here a little bit past day 40, certainly by day 50, most of those organ systems are identifiable histologically. So this is a time of very rapid organ growth, and it's a time of exceptional vulnerabilities to nutrient deficiencies or any other sorts of stresses. Oftentimes, we would see those vulnerabilities as livestock production people as just uh, lost pregnancies or lack of, we might even identify it as lack of conception, depending upon when that occurred. So we've done a series of studies I want to share with you here today. And the first one, our overarching hypothesis was that a moderate maternal nutrient restriction during the first 50 days of gestation in beef heifers would impact nutrient supply, placental development, and gene transcript abundance in the fetal liver, muscle, and cerebrum, or the brain, thereby programming susceptibilities to impaired postnatal life outcomes. And so at the end of the day, what we really wanted to know is, does moderate nutrient restriction during the first 50 days of gestation in beef heifers actually matter to the offspring? Briefly, our experimental design used crossbred heifers. They were 16 months of age. They were assigned to treatment at time of breeding, and samples were taken at day 16, 34, and day 50 of gestation. We used a control treatment, which was 100% of NRC requirements to gain, and we used, and that was a 0.45 kilogram per head per day was the target. Our actual out to day 50 was 0.51 kilograms per day. And we had a restriction, which was a 40% global nutrient restriction accomplished by reducing intake to about 60% of control. And we were targeting maintenance. Our actual intake was about 0.08 kilograms per day. We then took tissues surgically by ovariohysterectomy, which was a technique that we developed and allowed us to get exceptionally fresh and good tissues. So they went immediately to the laboratory for dissection and workup. We took fetal liver, fetal brain, and fetal muscle at day 50 for the study I'm going to show you. There was day 34 and day 16 for some other parts of the work, but we're going to focus on these day 50 tissues because we could actually get these tissues at day 50. Not a lot of them, but accessible. And so this table's a little busy, but I just kind of want to focus you on this. This is functional categories for differentially expressed genes involved in tissue metabolism, accretion, and function. And it's presented as upregulated or downregulated from restricted compared with control heifers. And so let's just look at the yellow highlighted on your left. We have liver, fetal muscle, and cerebrum. Then if we look under liver, we have metabolic pathways, protein kinases, some other issues. There are some other pathways there. Fetal muscle, under that we have skeletal muscle, embryogenesis, and under cerebrum we have metal bonding and hippocampus and neurogenesis. The point I want to make with this slide, and one of the really unique things about this data set, is that in most cases, these genes were upregulated in those fetuses coming from the mothers that were in the restricted nutrition diet. Lots and lots of upregulated tissue, upregulated genes. And so our takeaway from this study, and I only showed you a little bit of data, we got many papers published on that study. One, heifer growth trajectory was altered, of course. Uterine placental vascular development was reduced. That data is published, didn't show it to you. Physiological fuels provided to the conceptus were changed. Gene expression was upregulated in fetal tissues from restricted heifers. Changes in methionine and homocysteine suggested epigenetic mechanisms were at play. And we concluded that additional work is needed on mechanisms and strategic supplementation, particularly with one carbon metabolites targeting the methionine and folate cycles. And so just briefly, we have the methionine cycle highlighted in blue and the folate cycle highlighted in yellow, orange over here. One's on the left, one's on the right. They overlap here, of course. Most of you all are familiar with this pathway. When we went to think about and consider targeting one carbon metabolism, we wanted to look at the methionine cycle and the folate cycle, the transsulfuration pathway, and we wanted to look at nutrients, methionine, B12, choline, and folate, asking the question, can we provide more of that in a time where one or more of these nutrients might be limiting for critical methylation that might impact developmental programming events? Can we mitigate those? And so we wanted to provide some of these. We also tracked homocysteine in our results. So we did a follow-up cell culture study knowing that these were things we wanted to look at. And so we did an epigenetic modifier supplementation, and we wanted to know if it would improve mitochondrial respiration rates and alter DNA methylation of bovine embryonic fibroblasts. That data is published about a year ago, and the answer was yes, it can do that. Our hypothesis when we went to study this was supplementation of OCM the four that I mentioned, methionine, folate, choline, and B12, to bovine. Now, this is a cell culture study, and for whole animal physiologists, this is kind of going out on a limb for us, but we needed to take a first look in something that wasn't too expensive. So we used bovine embryonic tracheal fibroblast in divergent glucose media, so adequate nutrition or low nutrition was our thinking about that. And our objectives were to determine if supplementing OCM to bovine embryonic fibroblasts cultured in these divergent nutrient supplies would improve cellular growth as measured by proliferation rates and total cell counts. So we sourced the cells. We did a 2x4 factorial arrangement, 2 levels of glucose and 4 levels of 1-carbon metabolites. And we did 1-carbon metabolites at 2.55 and 10x, and of course our baseline control. We limited methionine to 2x across all treatments because it can be cytotoxic. So we just capped that one. In our analysis, growth rate and cell proliferation and mitochondrial respiration. We analyzed transcript abundance of methionine and folate cycle enzymes and DNA methylation by reduced representation bisulfite sequencing. So we took a really hard look at this study. So real quick, labeling index here. Across treatments are across the bottom, and proliferating cells are on the left side of your slide on that axis. And we can see we got considerable differences here with low in the green declining and the high increasing in response to OCM supplementation. Very different responses, whether they were getting low or high nutrition to our treatments. So if we look at growth rates from hour 1 to hour 72 in these cell lines, the first thing that we can see is we have a quadratic effect in our low treatment, with the 5x seeming to be the best. If you look at the high, we have a cubic effect, and we can see that plotted out by the line. So we're getting responses to our OCM treatment, and the responses are different depending upon how much energy is being supplied. So next, we looked at reserve mitochondrial capacity using seahorse analysis. That's shown here. And we can see that we have the low in green again and the high in yellow. And those responses are different. And I just want to take those reserve mitochondrial capacity responses in the upper left-hand part of your slide. I want to take those lines and laying over the growth rates and take a look at how those look. Those responses are exceptionally similar, and it would appear that reserve mitochondrial capacity be very related to total growth rates as we measured them. So our results from this cell culture study was that total cell growth was greatest in the high 10x mitochondrial maximum respiration, and reserve capacity was greatest for the high 2.5 and the 10x compared to all other treatments. We also would find in low cells that the total growth rate and mitochondrial maximum respiration and reserve capacity increased quadratically, and that the biological processes identified due to differential methylation included the positive regulation of GTPase activity, molecular function, protein modification processes, and phosphorylation and metabolic processes. So we came away from this cell culture study going, we really probably need to move this into some large animal work. We interpret our data to imply that OCM increased the growth rate and mitochondrial function beyond the control treated cells in both the high and low cell treatments, which may be due to changes in methylation of genes involved with growth and energy metabolism. So evaluating responses of OCM supplementation to dams at different energy intakes is warranted. It's what we concluded at the time. We still hold to that conclusion. It is warranted to identify whether embryonic responses in vivo are similar responses are observed in vitro. So Cell growth and proliferation in cultured embryonic cells in vitro may be directly attributed to increases in mitochondrial efficiency. Cellular requirements for OCM may be dependent upon energy availability, and further studies in vivo should be designed to target strategic supplementation during early gestation in beef cows. So to that end, we did design a couple of studies here, and I want to share some of that data with you. This is a little bit busy, but just hang with me over here. On the left, we have breed and assigned to nutritional treatment, N equals 144, day 0 breeding, far left of the slide. We then have 4 treatments, control minus an OCM treatment, same OCM treatment as before. 1 carbon metabolite modifiers, we're looking at 4 of them in combination. Second treatment was control plus OCM. Third treatment was restricted minus OCM, and the fourth treatment was restricted plus OCM. Then, as we moved out, we started those treatments at breeding, and we've taken tissue samples at day 63 and also at day 161. And we have another study on the ground where we hope we'll put those calves on the ground and look at post-parturient responses. And so we did design this. We had a control and restricted. Our control was targeted at 0.45 kilograms per day, and our restricted was targeted at a minus 0.23 kilograms per day. So some data from this study. This is from day 63. It's 1 carbon metabolite supplementation. The effects on maternal liver folate metabolites as determined by mass spec analysis in collaboration with the USDA MARC. And some takeaways from this are that we see that we have 5,10-methylene and methylyl tetrahydrofolate responses to gain. And I just draw your attention maybe over here to the p-values for gain and supplementation, where we got large number of differences in the metabolites. And the bottom line here is that our supplementation of OCM was being widely responsive in folic acid metabolites in the maternal liver. So our treatment was working on the maternal side. If we look at oxygen consumption rates and respiratory ratios in the isolated mitochondria from the fetal liver, from the mothers receiving these treatments at day 161, we see that we have interactions between our gain treatment and our OCM treatment in mitochondrial respiration. In each of those cases, control heifers receiving the plus OCM treatment had fetuses that had greater respiratory rates in the liver mitochondrial than the other treatments. It's an interesting observation. Supplementation of these 1 carbon metabolites or micronutrients are have an effect on the offspring mitochondria in utero at day 161. In another study that we've worked on, I got a little bit of time here and we'll share a little bit of extra data with you. This one was entitled Strategic Supplementation and Rate of Gain in Heifers. And our strategic supplementation this time was a vitamin and mineral supplementation, and we had differential rates of gain during the first trimester of gestation. So we went out to 83 days of gestation, and the data I'm going to show you from this study is focused on fetal liver amino acid, carbohydrate, and energy metabolic profiles at day 83 of gestation. The objective of this study was to evaluate the effects of feeding heifers a vitamin and mineral supplement and targeting divergent rates of gain during early gestation on fetal liver amino acid, carbohydrate, and energy profiles. To do this, we had 72 crossbred Angus heifers, and they were randomly assigned to a 2x2 factorial arrangement to one of 4 treatments comprising the main effects of vitamin and mineral supplementation and of feeding to achieve different rates of gain. And our rates of gain targets in this case were 0.28 kilograms per head per day versus 0.79 kilograms per head per day. Both very reasonable rates that we would see in production practices. Just a snippet of some of that data. You have the treatments on your left. You see at day 83, we collected those tissues. We did metabolomics of fetal liver collected at day 83, and some summary of that data. This sort of big data work creates lots and lots of pathways, and trying to get that summarized down can be a challenge sometimes, but I think this works pretty well. If we start up at the top, we certainly had a difference in fetal biparietal distance. As we move across, branch chain amino acid abundance in the liver was different. Fetal biparietal distance is a physical measure that we did. It's not in the liver. But pentose phosphate intermediate abundance was lower in no VTM versus our vitamin trace mineral supplement. Oxidative phosphorylation metabolites were lower in our no vitamin trace mineral supplement compared with our vitamin trace mineral supplement, and TCA cycle intermediate abundance was lower in no vitamin trace mineral compared with vitamin trace mineral supplements. Large changes in metabolites in these fetal livers at day 83 in areas that matter to us from a production standpoint. TCA cycle intermediates, oxidative phosphorylation, pentose phosphate intermediates, branch chain amino acid abundance. Those are important attributes in our metabolism of developing fetuses and an offspring on the ground. The results from this study demonstrate that a majority of metabolites that are affected by rate of gain or vitamin and mineral supplementation are decreased in heifers on a greater rate of weight gain or a greater rate of vitamin and mineral supplementation. Our findings also supported greater metabolic activity in fetuses from heifers on an increased rate of weight gain and supplemented with vitamin and mineral compared with those on a slower rate gain or not supplemented with vitamins and minerals. The results show that fetal amino acid, carbohydrate, and energy metabolism can be altered by maternal nutrition during the first trimester of gestation. Now, this is metabolism in those day 83 fetuses in their liver. The data also suggests that these changes manifest in altered fetal size and calf birth weights. Question, would there be measurable changes to feed, growth efficiency, or reproductive efficiency in the heifer offspring? Are we altering intake? Are we doing those kinds of things? That kind of work probably still needs to be done. We may have some of it, but it's not published yet and not ready for show and tell. Current and emerging future efforts. We're interested in strategic supplementation of OCM and other nutrients, transgenerational programming of outcomes, the microbiome and developmental programming. We're starting to do some things with that with our collaborator, Samat Amat and his team. We're interested in epigenetic regulation and modulation. Can we get in there and mitigate some of that and improve production efficiencies? What about periconceptional nutritional programming effects? And what about the bull? We have some work going on there too. Implications. Maternal nutrition during early gestation matters. Even moderate changes in nutrient supply have impacts. What are the specifics? We need to know specifics before we can manage it. We can't really manage what we can't or won't measure. That's important, and we need to figure out what we should measure, and then we can figure out how to measure it, and then we can manage it. What are the mechanisms? Well, we think we're down the right track on that, but there's a lot of work to be done. Can we manage developmental programming? Can we use strategic nutrient supply to manage developmental programming? And can we relate strategic nutrient supply to demand during early pregnancy? Can we even measure demand during early pregnancy? Data from research in this area are expanding our understanding of biology, impacting animal agriculture, and have potential broad implications on major societal issues. Currently, our knowledge of developmental programming is in what I would call the embryonic stage. This blue box at the bottom that has the word known in it is an idea of what we know or what's currently known about developmental programming. This orange box is a representation of what we don't know. There's a lot more that we don't know than what we know, but we know enough that there's to know that there's a lot of really cool information we need to understand out there, and it likely impacts livestock production and developmental outcomes to a large degree. So, early maternal nutrition matters. What are the specifics? What are the mechanisms? Can we mitigate negative and foster positive outcomes? This is a critical research need with broad impacts in animal agriculture and in broad society. With that, I need to acknowledge collaborators both at NDSU and other locations across the world. Without really good scientists to work with and collaborators that make things fun, it'd be difficult to get this kind of work accomplished. Also need to acknowledge USDA, National Institutes of Health, our industry partners, and North Dakota SBARE initiatives. But I certainly want to acknowledge the graduate students, undergraduate students, and postdocs that pour their hearts into this kind of work. We really, really need to acknowledge them, because without them, we wouldn't get hardly anything done. And with that, I'd like to say thank you and turn the floor back over to the moderator for questions if there are any.